Hey all, um, we're going to be going over experiment 1 for this video. It's media preparation and sterilization. And a lot of y'all are going to be recognize a lot of what's in this PowerPoint just from labs that you've done. But the purpose of this lab was to kind of go over the general parts of micro lab and how they kind of get to where we are. So, so the first part was cultures. Like you know already, you need to grow bacteria and stuff. You need to culture them, and if you want to grow them in large amounts, that you can actually observe useful data. And so, there's two ways to grow a culture. The first one is you can use a medium that has it's called a complex medium, in that you take something like meat or yeast and you just kind of grind it up and then put it in something, and then bacteria will grow on it. Effectively, they do more stuff than that, but for our intent and purposes right now, that's all you do. And the thing about these are that they can sustain life. Like, obviously you've seen stuff get infected on meat and then maybe change the color of meat or something, and then these are actually living organisms in and of themselves, so they can absolutely sustain life. The one drawback here is that you don't know exactly what is being used in this complex medium. And so another way to do it is to actually put in exactly what you need to grow bacteria. And so you can use what's called a defined medium, in that you can measure out like the amount of nitrogen you need, the amount of carbon you need, the amount of oxygen you need, the amount of whatever you need, sulfur maybe, and actually just and then put the chemicals in there that is exactly what you need to optimally grow a specific target bacteria. And so that's called a defined medium. And so you will need to know the differences between these two. Another way to look at something is, like y'all have seen already, you can look at slants, broths, blah, blah, a plate, and then a deep. Sorry, brain fart. Okay, so the good thing about slants is that they have a very thick level of nutrients right here, but also have the surface area to grow in a good amount of number. If you look at like a plate, it's the depth of the plate isn't very deep, and so you don't have that much growing. Like you can't really preserve these things as long as you can in slants. But the good thing about plates is that they have a very large surface area, and so they're really good for isolating bacteria. And then for deeps, they again, you can stab them. We use these for our flagella experiment, and the ones with flagella moved throughout the deeps, and then the ones that didn't have flagella stayed where we stabbed them. And so these are the two ways that you can describe medium. What you do is you kind of combine these, and so you have like a complex medium that is also a plate, or you have a defined medium that is a slant, or something like that. So you have like a complex plate medium, and then you have the broth medium, in that you can grow and measure the turbidity and other things as well. We'll get to later. The things about slants, plates, and deeps is they're all more have the consistency of jelly, as you've recognized, relative to the broths. That's because of the auger in there. And we'll be getting to that next. So the auger is made from red algae, It's called the phylum is Rhodophyta, which you will need to know. And read your lab manual to see why it is solidifying. It's because of the temperatures and the usefulness there. Um, but what's important about Auger is that we'll be using different types of auger throughout the semester. A big one that we use is tryptic soy auger, the TSA plates that you see, like the plates that are labeled TSA. Um, they're very good at just growing general bacteria. Um, and so, what you'll need to know is the type of auger and then what's in each one. So, and I mean, a lot of them, like, it's pretty self-explanatory. Like, you have a nutrient auger, it's like beef and then peptone, and like peptone is like a way to remember it is like peptone is like protein, and then you have a beef extract, and beef is very high in protein, and then all of them have auger in them. So it's not really that hard to remember. Tryptic soy auger, tryptone, soytone, sodium chloride, and auger. Like, um, so just learn those. It should be relatively not that bad. Um, okay, so for the next one is how to sterilize. So the big way to sterilize is autoclave, and you kind of just put the plates in here, and then you close off this, and it's very high pressure, very high temperature, and you sterilize that way. And the high temperature primarily kills the bacteria, but the pressure as well in combination can even kill sporulating bacteria. And again, you probably learned that in class. Another thing, how we inoculate. I mean, how do we sterilize inoculating loops? We flame them, and so that's called dry heat sterilization. And so you would put your inoculating hook right here, and then it'd burn and glow, and you know that it's sterile. Another one is hot air sterilization, which is pretty much like an oven. And again, you just put it in there and close it, and then the high temperature kills most of the stuff. The last thing is, or another one is filtration. So you have like a little filter right here that's just has really small pores, small enough to where chemicals can get through it, but big enough to let chemicals through it, but small enough to allow bacteria to be filtered out. And so you have this nice little setup in that you have like a syringe, and then you put your little thing. I think she might have passed around one of these if she got there in class or not. But yeah, that's how you filter sterilize stuff. The next one is using gaseous chemiosterilizers, and what you do is the big one here is ethylene oxide. If you're in organic chemistry, what happens is that you have something does an SN2 reaction on the backside of this epoxide, and which if you're an organic one you probably haven't gotten to yet, but what this means is that this ethylene oxide is very reactive with things, and so because of its reactivity, it actually kills a lot of bacteria that way. And so this allows for stuff that's like heat sensitive, like the plastic petri dishes we use, and the plastic broth cultures, and the plastic syringes, and anything that's plastic a lot of the time gets ethylene oxide gas. UV radiation. So we did our UV experiment, right? And you saw how 5 minutes can kill a lot of the bacteria on a plate. And so if you do something for maybe even a longer time, you can sterilize things that way. And then this is just another form of radiation, in that you have like ionizing radiation, in which you make an ion, and then that ion reacts very heavily with things. And so that's just a general overview of that. For experiment 1 especially, it's a lot of just conceptual based, so make sure to read the background information, and then try to, what I did, I got a pretty good grade on the midterm and final, and the way I did it was I just pretty much memorized well, not memorize, but learn the theory, but then effectively memorize like the pages. And so what I would do is I would read a paragraph and then take note of what each sentence is saying, and then close my lab manual and then repeat that paragraph back, and then do that until I could say the entire background information without stopping. And I just redid it over and over, probably like 10 times. And it's a lot, but it was very successful in my endeavors. And so that's what I'm going to recommend to y'all, but I don't know. But I'll get to experiment 2 will be a lot more conceptualization stuff. So read the lab manual for experiment 1, and I'll see y'all later.